ركزوا معي الله يرضى عليكم لأن المحاضرة دنس بمعنى الكلمة. It's fun though. So هنتكلم على السيل سيجنالينج. طيب حلو. سيلز لازم إنه يصير communication بين بعضهم ومع السراوندينجز وحتى داخل السيلز نفسها. زي ما احنا بنعمل communication مع بعض وعندنا different modes of regulation. مرات بكون different modes of communication. مرات بكون verbal، مرات بكون بالعيون، مرات بكون تلامس، okay, and so on. برضه cells عندهم different modes of communication. فعندنا cell interaction. إنه حكينا مثلا الإيكوهيرنس بصير interaction ما بين السيلز مع بعض، وكيف إنه بيكونوا connected مع الانترميديت filaments أو بيكونوا connected مع الأكتين cytoskeleton. فهذا يعني direct cell interaction. في عندنا paracrine signaling, basically sending an email من cell to another cell. So a molecule would be sent or would be released. A molecule would be released from one cell to the other. وهذا molecule would bind to a receptor على cell surface of the other cell, and the signal is sent inside. Just like really sending an email. هذا بنسميه paracrine signaling. Paracrine signaling. مرات بنبعث احنا messages لحالنا. أنا for example I have my own WhatsApp group. مش لأنه أنا lonely، بس لأنه أنا ببعث شغلات لحالي. Articles، مش reminders، يعني برضو عندنا reminder هذا الكالندر. We send يعني a message to ourselves sometimes. هذا بنسميه autocrine signaling. Okay? A cell would send something, a message, to itself. وعندنا endocrine signaling, whereby a hormone, that's what it's called, a hormone would be sent, released from one cell. It goes into the bloodstream, travels a long distance, and it binds to a receptor على cell surface of a faraway cell. هذا بنسميه endocrine signaling. كثير منيح. هلا ايش السيجنالز اللي بتنبعت؟ different types. ايميل، phone call، مسج عادية، SMS، مرات بتكون ميديا، مرات بتكون written message، whatever. برضه السيل عندها different modes of messages that it can send. ممكن تكون المسج عبارة عن peptide، أو ممكن تكون large peptide، polypeptide، a protein. فممكن يكون for example insulin، glucagon، whatever. neuropeptide تشتغل على النيرفس سيستم. ممكن يكون small molecules amino acids، neurotransmitters معينة زي serotonin for example. حكينا على جابا، حكينا على شوية منهم هدول بالبايوكيمستري. ممكن يكونوا steroid molecules، يعني lipophilic molecules derived from cholesterol مثل الكورتيزول، مثل الاستروجين، الاندروجين and so on. ممكن يكونوا eicosanoids that 20 carbon lipid molecules derived from arachidonic acid. برضو حكينا على بعض هدول as well. وممكن يكونوا gases like nitric oxide, carbon monoxide and so on. ماشي. So lipophilic hormones من الاسم lipophilic, so they are hydrophobic. هدول الهرمونز are mainly derived from cholesterol. And if you look at the structure of these molecules, بتلاحظوا انه they really look like cholesterol modified. Hydroxyl group هون, ketone group هناك and so on. Of course I don't want you to memorize the structures. Lakin, we have examples like testosterone, estradiol, progesterone, cortisol, aldosterone. Remember the names. Remember the names. لأنه راح تدرسوهم next semester. All of these molecules وكيف يصير لهم synthesis. How do these hormones function? Mainly intracellular. هلا راح نعمله احنا خلال هاي المحاضرة انه راح نشوف هذول molecules, how do they behave? How do they send a signal? They all do so by binding to a receptor. هذا الريسبتور once it binds للligand, ligand يعني small molecule بيبعت signal, ligand. Once the receptor binds to the ligand, a receptor sends a signal. كويس؟ Okay. Now, how do lipophilic hormones function? لأنهم small and لأنهم lipophilic, they diffuse through the cell. They don't need a channel. They don't need a carrier. They can just diffuse through the plasma membrane. And they bind to a receptor. This receptor is intracellular. داخل السل. This receptor is virtually cytosolic. يعني it's there. It's free. It's bound to a heat shock protein. اسمه heat shock protein, HSP. This heat shock protein prevents the receptor from being active. Once the hormone binds to that receptor, the heat shock protein is released بفلت واحد اثنين. The receptor that is bound to the hormone dimerizes, يعني it forms a dimer, and it gets translocated into the nucleus. بدخل على النوكليوس and it binds to the DNA. When specific places. We'll talk about them in the molecular biology course. And it controls gene expression, يعني gene activity. Some genes would be activated, other genes would be inactivated. And this causes an effect on cell behavior. The cell would behave in a certain way. It grows, it dies, it moves, it does whatever it does. It depends. It develops, it differentiates. وهيك بيشتغل reproductive hormones, sex hormones, androgens, estrogens, they induce certain effects. Differentiation, male, female. Cortisol, metabolism, for example. Bone resorption, and so on. Right? So this is how these hormones, ligands, act by an intracellular receptor. Excellent. Okay? They diffuse, they bind to the receptor, the heat shock protein is released, they dimerize, the receptor dimerizes with another polypeptide, another protein of its type. It goes into the nucleus, it binds to DNA. It controls gene activity, gene expression, and so on. Theory-wise. So this lecture will focus on cell surface receptors. We're not going to talk about these intracellular receptors. So, the way these ligands, these signaling molecules, the way they function, they bind to a cell surface receptor. They cannot diffuse through the plasma membrane. The receptor is at the door. Okay? They knock on the door. And مين مين انت؟ صح؟ مين انت؟ انا مش شايفك. Anyway, so and the signal is sent inside. So we have a ligand, the signaling molecule. It can be a hormone, it can be a growth factor, it can be a cytokine. نحكي عن cytokines شوية. Then, so they bind to a receptor on the cell surface, and the signal is transmitted. The receptor can be a G protein coupled receptor. It can be a receptor tyrosine kinase. We're going to talk about all of these. Different types. And the signal is transduced. It is sent to a transducer. Okay, ما بدهاش يعني intelligence عشان نحول الكلمات. A transducer, a transmitter. Okay, what it does, like G protein, like RAS. We're going to talk about all these. هدول, they just send a signal. They do not do any action themselves, other than sending a signal. They have to letter from one person to another. That's it. That's all they do. And lamine to an effector molecule. The effector molecule does an action, amplified action. It's mainly an enzyme, like adenylate cyclase, like MAP kinase. And the effector, this molecule, can act on other effector molecules, other enzymes. ماشي? You can have in some signaling pathways. Pathways يعني نتكلم عن الطريق. لأنه إيش؟ إحنا بنحكي على طريق من 1 ل 2 ل 3. You can have secondary messengers. اوكي، الولد الصغير اللي بنبعته عشان يجيب لنا باكيت سجاير، أعوذ بالله. أو كيس سكر، whatever. اوكي، أو روح عند الجارة أجيب صحن مش عارف إيش. الولد الصغير هذا. هذول السكندري messengers. ليه بنبعت السكندري messengers؟ لأنهم فايعين. They're overactive, hyper. روح اركض. هذول السكندري messengers. That's what they do. Like calcium ions, cyclic AMP. And then we have the final target molecule. It can be DNA. It can be an actin binding protein. Okay. It can be a channel. It opens up, it closes down, whatever. And then you have the response. Based on that, you have a response. Response, cell division, cell death, cell proliferation, cell differentiation, movement, whatever. Then you have an action. We're going to talk about all these. Sometimes, you can have a response that is primary, or you can have a secondary response. And sometimes you can have a tertiary response. Okay, so this signaling pathway, the final result may not be what the cell wants, but it's something like to activate another pathway or another action. So for example, you can have a signaling pathway that results in expression of a certain gene. This gene would activate other genes. So you have a primary response for the first gene. Now the other genes are the secondary response. So the whole pathway is to activate these other genes. Sometimes these other genes can activate another set of genes. So that's a tertiary response. Okay? اسمعي يا جارة والحكي لك يا كنا. مش هيك بيحكوا؟ Okay. So the whole purpose is not really to send a signal to this person or this molecule, rather it's the other molecule. And So this lecture, yes, I'm going to talk about some things that are specific. But the whole point is for you to understand the concept. The whole chapter in the book is full of examples. Yeah, I want you to know these examples. لأنه أنا راح نحكي على هدول examples later on. We're going to talk about all these examples later on in other courses, not necessarily in this course. Okay, so it's good for you to know. Yes? Okay. So the idea here is that you have a primary response. So you have a signaling pathway, A to B to C to D, and then you have expression of a certain gene. Okay? Other gene would activate expression of other genes. So the primary response is to express this one gene. The secondary response is when this gene activates expression of other genes. This is called secondary response. No, it's different. It's different. Okay. Hello. So let's go through some examples. G protein coupled receptors, G proteins and cyclic AMP. We talked about this pathway in biochemistry. What are G protein coupled receptors? They are 7. It's a family of receptors. It's not only one receptor. Family. They have هدول receptors. They are transmembrane proteins, and they have 7 transmembrane domains. So they are inserted inside the membrane 7 times. In, out, in, out, in, out. زي الزر لما نخيطه. In, out, in, out. Okay? 7 transmembrane domain proteins. They have a cytosolic site, cytosolic site, and an extracellular side. The extracellular site is where the ligand binds. When the ligand binds, the receptor shakes. It causes, the whole thing shakes, it causes the cytosolic site to change a little. بتغير شكله, structure تبعه changes شوية. Okay? هذا اللي بيبعث السيجنال. So, هدول receptors are really important in our body, physiologically. They make up a large proportion of receptors in the body. They control hearing, they control taste, they control vision, they control cell proliferation, هدول receptors. There are so many receptors in our body. And they are they make up 20 targets for 25% of drugs. هذا percentage, and I read it like 15, 20 years ago. Maybe it's different, and they make up in main targets of drugs. Right, so these are the receptors. هلا. Why do they call them G protein coupled receptors, GPCR? Because they are coupled مع G proteins. Okay, من النيم. G protein coupled receptors. ايش هم ال G proteins? G proteins are the ones that we're talking about behind the pathway. هذول, they are heterotrimeric G proteins. So they are made of 3 polypeptide chains, alpha, beta, gamma. They are different from each other. They are heterotrimeric proteins. G proteins because they are regulated by binding to GTP. Again, عنهم كتير. أنا بعرف إنكم خلاص we got bored of these proteins. They are important. So these G proteins, alpha, beta, and gamma, they associate with the receptor, the G protein coupled receptor, when the receptor is bound to a hormone, ligand. The cytosolic site changes. Now they can bind to the receptor. When they bind to the receptor, the alpha subunit is connected to GDP, associated with GDP. When they bind GDP, and I hate this by the way, لأنه مش واضحة. The idea إنه GTP comes in, it kicks out GDP. هي actually, when the G protein binds to the receptor, GDP dissociates, and GTP comes in. Okay, it's not GTP أول, then no. GDP comes out, GTP binds. This causes the release of alpha. I think it causes the release. By the way, what I'm going to say is written in this slide. You have the release of GDP. GTP binds, and the alpha subunit dissociates, تفلت من البيتا and gamma. Then the alpha subunit can bind to a transducer. The transducer, as an example, is adenylyl cyclase. They knew before, or they said before, that beta and gamma are the only function of beta and gamma in whom they are inhibitory of alpha. Like, it turns out that beta and gamma can, once they dissociate, they can bind to other enzymes and other transducers. They have their own mechanisms of action. They have their own thing. But we'll focus on alpha. So alpha binds to an enzyme. And this enzyme is now activated. For example, you have adenylyl cyclase. What adenylyl cyclase does is that it converts ATP to cyclic AMP. Hell, before going into adenylyl cyclase, the alpha subunit must be inactivated. It has to be inactivated, otherwise the signal is always on. How does it get inactivated? By hydrolyzing GTP to GDP. And this happens by an intrinsic GTPase activity of alpha. Intrinsic يعني ايش؟ يعني internal، إشي داخلي خاص بالألفا subunit. It is an enzymatic activity that acts on itself. It doesn't act on other proteins. Okay, من الداخل. So GTP is ايش يعني؟ إنه it breaks down GTP, like protease, peptidase, saccharidase, lipase, okay, and so on. So this GTPase activity hydrolyzes GTP to GDP, and alpha becomes inactive, and it associates with the beta and gamma again. And it waits for another signal, ligand binding to a hormone, cytosolic site changes, it binds to the cytosolic site, and the same cycle. There are regulators for the GTPase activity. They are called GTPase activating proteins. ماشي? They facilitate, they speed up the intrinsic GTPase activity. يبقى عندنا هدول الريجوليترز. There are also regulators for the release of GDP and binding of GTP. يعني there are activators of the G proteins and there are inhibitors for the G protein. The inhibitors are GTPase activating proteins. The activators are known as guanosine or guanine nucleotide exchange factors. They facilitate, they speed up the release of GDP and the binding of GTP. Okay, GEFs, they are known as GEFs. Oh, For these specific G proteins, they are known as regulator of G protein signaling, RGS proteins. Regulators of G protein signaling, RGS, and there are several of these proteins. Yes? So it's a cycle really. يعني إنه في عنا the release stimulated by the GIFs by the RGS, and then we have the gaps. مكتوبة somewhere here. معلش أنا حضرت محاضرة الساعة 11:30 بالليل. أوكي يلا اصحوا عشان تعرفوا.
دكتور
نعم.
الRGS نفسهم gaps.
الRGS الRGS well they can be هم usually الجابس هم inactivators والRGS هم الجافs exchange factors. لحظة أشوف معلش. مش مكتوبة actually هون. Usually the RGS, regulator of G protein signaling, are activators. They exchange GDP for GTP.
And GIFs. And GIFs.
And the gaps. Yeah. Gaps are the inactivators. Okay, خلاص؟ ماشي. ما لاحظوا؟ أمورنا تمام؟ Lara, are we doing well? Yeah, bismillah. So let's talk about the secondary messengers. إيش هم secondary messengers? Taken from the book. It's a compound whose metabolism is modified as a result of ligand receptor interaction and functions as a second so by regulating other intercellular processes from the book. Okay. So it's the first the first messenger is the hormone itself. So that's why they call them secondary messengers. And the hormone is the primary messenger. Usually these are small molecules. الولد الصغير الفايع هذا hyperactive. So like cyclic AMP, it can be calcium ions, it can be cyclic GMP. So how is cyclic AMP produced? From adenylyl cyclase. This is ATP. It takes ATP and it forms a cycle, a ring structure. That's why they call it cyclic AMP. And we have the release of two phosphate groups. هذول phosphate groups are known as pyrophosphate, just to let you know. Okay, PPI. إيش يعني I? Inorganic. Okay, right. In order to terminate the signal, to stop that signal, the cyclic AMP must be removed. How does it get removed? By another enzyme known as cyclic AMP phosphodiesterase. It converts cyclic AMP to AMP. Now the signal is terminated. Okay? An example, how does cyclic AMP function? Well, what it does, again, you have adenylyl cyclase, activated cyclic AMP. Cyclic AMP binds to effector proteins, like enzymes, primarily, enzymes, like protein kinase A. Protein kinase A, once it binds to cyclic AMP, it gets activated. And it's a kinase. It phosphorylates other proteins. And that's how the signal is transmitted. Some of these proteins that are phosphorylated are effector molecules themselves, like enzymes, other enzymes. So you have an enzyme that activates other enzymes, or it can inhibit other enzymes. And you have these enzymes that can activate other enzymes. So these are all effector molecules. The whole thing, we call it a signal transduction because a signal is sent from one molecule to another. Signal transduction. Okay? We talked about protein kinase A in biochemistry. Protein kinase A has so many effects, and some of these effects are cell specific. So it can function in muscle cells, and the outcome is different, and it can function on liver cells, but the outcome on liver cells is different than muscle cells. Muscle cells, contraction, okay, metabolism of some sort. Liver cells, well, mainly metabolism. Okay? An example is glucagon, the hormone. It binds to its own receptor, G protein coupled receptor. You have activation of eventually adenylyl cyclase, then cyclic AMP, then you have protein kinase A. Protein kinase A can act on glycogen synthase or glycogen phosphorylase. An enzyme acting on enzyme, acting on enzyme, and so on. And then you have whatever effect. Glycogen synthesis or glycogen degradation. The catalytic subunit, and once you have binding of cyclic AMP to protein kinase A, the catalytic subunit is released from the inhibitory subunit, the regulatory subunit. هذه حكيناها في البايوكيمستري as well. This catalytic subunit can phosphorylate other enzymes, protein, glycogen synthase, glycogen phosphorylase kinase, whatever, all of these, we talked about it before. Or it can go into the nucleus. It's free. It was trapped. لأنه it's bound to the regulatory subunit. It's inhibited by the regulatory subunit. Once it gets released, it can act on enzymes or it goes into the nucleus. And it phosphorylates. It's a kinase. I mean, all it does in life is it phosphorylates. It can phosphorylate a transcription factor. شو هم transcription factors? Proteins that regulate gene expression. Transcription, production of messenger RNA. ماشي? So yeah, it can act on gene expression. Some of these, so it can act on a transcription factor called CREB, CRE binding protein. We're going to talk about these later on. Okay? By inducing gene expression. Expression of what? What genes? That do metabolism. It activates, for example, the glycogen synthase by phosphorylation. But when it goes to the nucleus, it activates this transcription factor. ايش راح يعمل? Produce more glycogen synthase. يعني you have really quick effect اللي هو على طول على glycogen synthase, and you have a slow action اللي هو gene expression لأنه it takes hours production of messenger RNA, بعدين translation, production of the protein, وبعدين يلا transport this protein out of the ER, Golgi, whatever. All of this modification takes hours. Okay, like in phosphorylation of protein kinase, phosphorylation of glycogen synthase, it's quick. So, you have target proteins that are phosphorylated by protein kinase A, and this signal must be terminated. Pay attention. When you have a signal that is activated, it has to be terminated. Otherwise, it's going to keep going on and on and on and on. Keep it. So, the protein that gets phosphorylated by protein kinase A and it gets active, how is it inactivated? By phosphatases. So what the phosphatase does is it removes the phosphate. So if this target protein is activated by phosphorylation, it has to be inactivated by dephosphorylation. فهون بنتكلم على phosphorylation by protein kinase A, dephosphorylation by phosphatases. ماشي كويس؟ معي؟ أمور تمام؟ طيب hear me. Why do cells use effector enzymes? Why do did they use them? For several reasons. هذا مش موجود بالكتاب. It's easy and quick regulation. Phosphorylation, dephosphorylation. It takes less than a second. Activate, inactivate, really quick. Okay, and it's easy. Just reversible covalent modification. Really quick. Binding to small molecules like cycling him, really, it's really quick. Just it binds. And that's it. It's sensitive. يعني the enzyme gets really active. Yeah, whoa. خصوصا if it is an allosteric enzyme. Why? Look at this. This here is the plot showing enzyme activity, the activity of an allosteric enzyme. يعني allosteric, it goes through a cycle of being on, off. It changes its structure. It has a plot, an enzyme activity plot that looks sigmoidal. It looks like an S. Now notice, little change in substrate concentration, or the activator, whatever. Look at this. Just little, wallahi, little change in the concentration of the substrate. Notice the change in activity. It goes up so much, or little increase in substrate concentration. It goes down really quick. That's why it's allosteric. It's very sensitive. It goes up and down, active, inactive, really quick. The other thing, you have an amplification effect. So you have an enzyme that can activate, let's say, 100 enzymes. Each one of them can activate 100 other enzymes. Yani 100 times 100, 10,000 enzymes would be activated in less than a second. Okay? By the way, how many biochemical reactions occur in our body per second? In our body, all cells. How many biochemical reactions per second? Maybe I said it before. 10 billion reactions per second in our body, in all of our cells. And they're all regulated. Amazing. So you have an amplification effect. Okay. Why are secondary messengers used? الأولاد الفايعة الصغار. لأنه they can be stored, like calcium ions. They are hidden. They can be stored in the plasma membrane. They can be stored in the endoplasmic reticulum. And then they are released. So they can diffuse from one compartment, plasma membrane, endoplasmic reticulum, to another, to the cytosol. From the cytosol to the mitochondria, and so on. So they can diffuse. They can move from one compartment to another. Calcium ions, diacylglycerol, phosphatidyl inositol 3-phosphate. Just examples. We're going to talk about these. The signal can be amplified because once you have this cyclic AMP, it binds to a protein kinase A. Well, again, you have amplification. The calcium ion can bind to an enzyme. It can be released. It can bind to another enzyme. It can be released. It can bind to another enzyme. So it can be amplified, the signal. And the other thing, different signaling pathways can cross-talk. Yani you can have calcium ions released. They can activate this pathway and that pathway and this pathway. And you have different enzymes that depend on calcium ions. So you can activate several pathways at the same time by secondary messengers. روح دق على جارات كلهم إما محمد وأم مش عارف إيش لا بروح من الطابق الرابع بنزل على البيسمنت. Okay, so that's what it does. Amplification, signal amplification. So this is what crosstalk means. Definition again from the book. طيب نعم.
In the crosstalk, هي نفسها تبعت primary or secondary responses ولا لا؟
No. Crosstalk means you have two signaling pathways pathways talking to each other. Okay, so you have again الولد الفايع. Okay, بدو على ام محمد وام محمود. ام محمد بتعمل action وام محمود بتعمل action تاني. Okay, so and then they cross talk. Okay, so sort of. So, هدول G protein coupled receptors. We got the concept, huh? So let's talk about the other receptors, enzyme linked receptors. It's basically a receptor on the cell surface that either has an enzymatic activity itself. The receptor has an enzymatic activity, a kinase activity, or it is bound, it is associated with an enzyme. Right? So we have two modes, مش moods. Okay? mood مزاج, mode طريقة. Two modes. We have receptor tyrosine kinases, meaning that it is an enzyme that is also a receptor. Oh, it's a receptor that is also a kinase. And we have non-receptor tyrosine kinases. يعني إنه they are bound to an enzyme, separate. And this enzyme can be released from the receptor. Why do we call them tyrosine kinases? لأنه we said in biochemistry that there are 3 amino acids that can be phosphorylated. Tyrosine is one of them. And the other 2 are serine and threonine. What do they have in common? Hydroxyl group. Protein kinase A is a serine threonine kinase. يعني it phosphorylates, it adds a phosphate group على serine and threonine. Now we're going to talk about tyrosine kinase. They phosphorylate, they add a phosphate group to the hydroxyl group of tyrosine. So you can have 2 types, either the receptor itself acting as a kinase, or you have a kinase that is associated to the receptor, non-receptor tyrosine kinases. Okay. That's the mechanism of action of receptor tyrosine kinases. The receptors are monomeric, one transmembrane domain, and you have 2 of them close to each other. The ligand comes in, like a growth factor. It binds to the cytosolic site. That gets the 2 polypeptides, the 2 receptors, to dimerize. So you have dimerization. ليه? Because connected by the growth factor. So you have dimerization. Now this, the cytosolic side, has the kinase activity. So this has a kinase activity, and this has a kinase activity. This kinase phosphorylates this receptor, and this kinase on the that receptor phosphorylates the other receptor. So we say we have autophosphorylation. The receptor phosphorylates itself. Okay? So we have binding of a ligand, then you have dimerization, then you have autophosphorylation. Autophosphorylation. Right. Okay? High mechanism of action. Questions? Okay. What does autophosphorylation do? 2 things. The kinase activity of the enzyme becomes even better. It becomes stronger. Okay? It gets activated or more activated. It's already active, like it gets more activated. The other thing, the addition of a phosphate group to the receptor allows proteins and other signaling molecules to bind. يعني صار نحكيها it creates phosphorylation, creates a docking site. Docking, D-O-C-K-I-N-G. شو يعني؟ يعني لما تيجي سفينة وبدها تصف بالمرسى نحكي انه صار في docking، انها صفت في مكان، وقفت في مكان، اوكي، ربطت وبعدين بربطوها بحبل، اوكي، docking site، مكان مش عارف بالعربي ايش docking اللي هو، اوكي، هذا الدوكينج سايت، انه فالفوسفيت جروب، شو أهمية الفوسفيت جروب؟ negative charges. في عندنا فوسفيت we have negative charges. هذه النيجاتيف تشارجز بتعمل electrostatic interactions with other proteins فبصير non covalent interaction. في عندي فوسفيت negative charges حلو نعمل electrostatic interaction فبصير في عندنا electrostatic interaction. So what happens is once you have phosphorylation of the receptor, now you have other proteins that can bind to that receptor. And the kinase activity becomes more active. So when these proteins bind to the receptor, the kinase of that receptor phosphorylates these proteins. Okay? So, واحد autophosphorylation بيعمل لي docking site. بيجوا proteins بربطوا على phosphorylated site. Once they do, the kinase activity, the kinase of the receptor phosphorylates these proteins. شو بيعمل؟ docking sites as well. The other proteins. احنا بنحكي على signal transduction، يعني ايش؟ يعني relaying signals. معلش انا كمان فسر الماء بالماء. هذا وجودي يعني ايش؟ انه relaying، انه بتبعت signal in molecule to molecule to molecule. هذا signal transduction. اوكي؟ فبصير انه السيجنال اخذها الريسبتور ببعثها لهذا المولكيول، هذا المولكيول بيبعث السيجنال لكمان مولكيول، بيبعث لهذا المولكيول and so on. So you have هدول proteins they localize to the plasma membrane, they can associate with other proteins فبصير عندنا cluster of proteins على الريسبتور. It promotes phosphorylation of these proteins by the receptor or other enzymes. And this stimulates the enzymatic activity of these enzymes. هلا نعطي examples. Okay. طيب non-receptor protein tyrosine kinases هذول ما عندهم kinase activity لكن they associate with kinases. Again, you have binding of a ligand. The receptor dimerizes. So you have these two coming in together. Allowing the two enzymes to be closer to each other. هدول الكاينيزز they phosphorylate each other. برضه نفس الشيء except إنه إحنا عندنا protein that is separate من الريسبتور أو إنه يعني أو okay they phosphorylate each other and then they can phosphorylate the receptor itself or other they can phosphorylate the receptor itself. So they first phosphorylate each other and then they phosphorylate the receptor. ليه بيعملوا phosphorylation of the receptor؟ نفس الشيء. Docking site. You have a phosphate group. هذه الphosphate group can bind to other proteins, other signaling proteins. Example, and I want you to know this example. The JAK-STAT pathway. The JAK-STAT pathway. Basically, it depends on here. It's active. High pathway is activated by cytokines. هتدرسوها بالimmunology. شو يعني cytokines? Small molecules, small proteins that signal for the control of growth and activity of immune cells and blood cells. طيب. هذول بيشتغلوا على immune system, cytokines, signaling molecules. In JAK-STAT, you have the cytokine binds to the receptor. Then JAK, which is a kinase, they get closer to each other. They phosphorylate each other, allowing for the binding of a protein STAT. STAT binds to the receptor, then Jack phosphorylates STAT. STAT is a transcription factor. Once it gets phosphorylated, it goes into the nucleus, it binds to DNA, stimulating gene expression. Yes? Okay? And that's what it does. هلا ايش هم الجينز? Well, it depends on the cell. It depends on the cell. Immune factors and so on. طيب آخر issue. Then we have, for example, integrin signaling. What I try to do إنه أحكي على شغلة لكن notice بحكي عليها in different lectures. Integrins, hemidesmosomes, focal adhesions. بعدين حكينا على the structure of focal adhesions in relation to the extracellular matrix أو in relation للactin cytoskeleton أو intermediate filaments وبعد and so on. Okay, هلا بنحكي على signaling. Now these integrin molecules, they bind to the extracellular matrix, and once they bind to the matrix proteins, you have على بالfocal adhesions, you have an enzyme اسمه focal adhesion kinase. It binds to the integrins. Again, you have phosphorylation of the receptor of the integrins. هلا عندكم docking site. بيجي هذا البروتين it binds to the receptor. Okay? And you have this protein, it's a kinase. What it does, other than it can phosphorylate docking site, it phosphorylates the receptor, it creates more docking sites for other proteins. نفس الشيء الاميونوجلوبولين superfamily حكينا عنهم قبل. Listen, I didn't record that lecture. With cadherins, they act in a similar manner. Okay? They bind to SARC, a protein known as SARC, بيصير phosphorylation. نحكي على هذه later on إن شاء الله. I'm gonna stop here. I'm gonna record this lecture again as well. يعني I had that one. معلش. الله معك. أي سؤال صحيح؟